All right, so hi folks. My name is Michael Robo, and today I'm going to talk to you about some of the uses that we've been doing with the GPUs on Big Red 200 for developing new methods to aid drug discovery. So a little bit about me. Prior to August, or I guess prior to September of 2022, I was a postdoc at the Indiana University School of Medicine. That's up in Indianapolis. I was joint between two professors, Jonah Vilsek. He's a comp chemist. His lab does a lot of work in developing new computational methodologies in drug discovery, as well as Tim Richardson, who is a medicinal chemist. And he was someone who I would sort of work with to sort of aid the experimental practice of medicinal chemistry. I still work with both of them as an IU affiliate, but in October, I moved to a new position at the Indiana Biosciences Research Institute, also in Indianapolis. Like I said, I'm still working with Tim and I'm still working with Jonah, doing many of the same stuff that I was doing in my postdoc, but now I'm starting to work on some other projects as well, and we sort of have more resources to maybe focus on other projects with practical applications as well. So just to give you an outline of what I'm going to talk about today, I'm going to give you the mile-high view. I'm going to tell you what you're trying to do on a drug discovery project. And then I'm going to tie that in with GPUs, how we can use GPUs to aid this process. I'll make this even narrower. I'll tell you why we wanted to use Big Red 200, why that was an advantage for us over other GPU resources that IU has, like Carbonate. And then finally, I'm just going to talk about some of the results of the work that I've been doing with the Vilsek group. So let's start with drug discovery, right? Let's say you have a disease. I am being general here because the process is fairly generalizable, and you want to develop a new treatment for that disease. The first thing that you're going to want to do is you're going to want to understand the biology of that process. That is a little bit out of my expertise. But to give you the short version, your body is filled with millions of tiny Rube Goldberg machines that we like to call proteins, and they all interact with each other in a very Rube Goldberg-like fashion with all these different pathways, and that pathway is going to have some kind of effect. Maybe it'll cause inflammation, maybe it'll cause dementia, maybe it'll cause erectile dysfunction. And what you want to do is you want to stop that process from happening. And the way you do that is you find one of those proteins in that signaling pathway, and you decide that you want to focus on that protein to try to figure out a way to shut that protein down, to inhibit it. And the most common way to do that is with what's called a small molecule. So small molecules are called small because they are much smaller than proteins or antibodies, which can also sometimes be used as therapeutic agents. I've put a couple of examples of molecules as well as the diseases that they're associated with below. Probably the two big advantages of using small molecules is that one, they're orally available. Patients don't really like needles, so if you can take a pill by mouth, that's going to be very helpful. And two, because they're small, they're also capable of going through cell membranes, which can act as a barrier to a lot of those larger molecules, such as full-on proteins. So this means that small molecules have access to a lot more targets that you wouldn't be able to access with something like a peptide. The way these guys work is, the goal is to shut this protein down. You can think of a small molecule as sort of a monkey wrench that you're throwing into the gears of that biological process. That small molecule is going to find that protein, is going to bind to that protein, and that binding event is going to cause the protein to stop doing what it was originally meant to do. So just to help motivate why we would want to use computational expertise in this process, I want to point out to you that, while testing small molecules can tend to be fairly cheap, in order to test them, you have to make them beforehand. And the synthesis process can be very expensive. So if you take a trained PhD organic chemist, and tell them to make a small molecule, typically you're going to be churning out small molecules at a rate about 1 per week. And the cost, when you include labor as well as the materials associated with that, is going to be about $1,500 per compound. So, these are pretty high costs, both in time as well as money. So the goal of a computational chemist in a drug discovery project is really to help prioritize what you want to do in experiments, to tell them these are the things that look best in the computer, right? Ultimately, it's a simulation, it's not reality. So there's never going to be that perfect correlation, but you're sort of loading the dice in the favor of the experimentalist so that they can be more effective with the time and money that they put into the synthesis process. So when you're thinking about how to use computers to aid drug discovery, it's probably worth just thinking about what you want that ideal treatment, what you want that drug to do in the first place, right? For starters, the goal is generally to have an orally available pill. In order for it to be orally available, that means that after you swallow it, your gastrointestinal tract has to be capable of absorbing that material. And once you've done that, it needs to be able to, go into the blood and distribute itself throughout the body so it can find that particular protein that you're looking for. You also need to worry about the processes that your body has to sort of deal with foreign chemicals. So you don't want, the liver to chew up that drug, and you don't want the kidneys to uptake that drug so that you can excrete it. And finally, you also don't want the drug to kill the patient. Toxicity is something that you want to think about too. And after you jump through all of those hoops, you then need to figure out how, right, is this drug actually going to be capable of binding to that protein and inhibiting it the way that you want it to be? So I sort of divided these into two groups. On the one hand is what's called ADMET, and the other hand I'm sort of calling, just a more general problem of potency. And, I'm not going to stand here and claim that ADMET is a solved problem in the field of pharmacology, because it's really not. But you have a lot of advantages in ADMET because every orally available drug needs to jump through all of those hoops. And that means that you have a lot of data about previous successful drugs that you can use to inform your predictions. So you have a lot of data points. If you have a lot of data points, that means you can create regression models. You can maybe even go ahead and use machine learning models. And that means that you can use generally just CPU hours to create pretty computationally cheap and reasonably accurate predictions about these different ADMET properties. Potency, on the other hand, generally doesn't have a lot of data that you're working on beforehand. And the idea is if you're working on a new treatment for a new disease, that's going to be a new target. So almost by definition, you're sort of working in this novel space where not a lot of people have done work in before. So can we hide these? Oh, we can. I guess we have to just choose the least. It's annoying. I can move over here for this. So that means that we can, because we don't have a lot of data that we're working with, we need to generate that model computationally. So this is really where the role of simulation comes in, where we can just start simulating at an atomistic level of what's going on during that protein binding process. There we go. So how do you determine potency in silico? Right? So the reason why small molecules are capable of binding to a particular protein is because, ultimately they want to. Thermodynamically, it's more favorable for these molecules to exist bound to that protein than it is for them to exist freely in solution in your blood. And that's going to be due to a lot of different interactions that the protein can offer that you can't get in solution. That'll range from like lipophilic interactions to hydrogen bonding events, etc. And these are the kinds of processes that you want to simulate in the computer. So what we do is we use this process called molecular dynamics that's going to simulate all of these different interactions. And we're going to use these molecular dynamic simulations to ultimately estimate the potency of a small molecule. So molecular dynamics, in a nutshell, you start with a set of equations that's called a force field, and that's going to give you a means to estimate the total forces that are applied to any particular atom in your simulation, according to this fairly complicated equation. We're looking at a lot of different things. We're treating atoms like balls on springs. We're sort of ignoring the quantum nature of molecular bonding. And we're going to look at bonds. We're going to look at angles, torsions, as well as long range effects like the van der Waals forces or electronic forces as well. Once you have that force field in hand, you can integrate the force over a really small time step and use that to figure out what the momentum on that individual particle is going to be. And then you can integrate over, again, a really small time step you can integrate the momentum over a really small time step to give you the position of what those particles are going to be. So this is not a perfect solution for the equation of motions for these particles, but given that we're going to deal with somewhere in the realm of 5 to 6 figures worth of atoms, that's going to be difficult, if not impossible to do. But as long as you work with a very small time step, you end up with a simulation that does a good enough job of approximating the true motions that you would expect to see for these particles. So that's really all that molecular dynamics is. You have your set of forces, you use your forces integrated over a really small time step to give you your momenta. You do that integration again to get your position. And then with those new positions, you recalculate what the forces are going to be, and you just loop that over and over and over and over again. The challenge is that you need to use a very small time step. A typical time step in molecular dynamics is 2 femtoseconds. But the simulation length that you want to go for tends to be much larger. In this case, a very common simulation length is 5 nanoseconds, right? So you can see there's a big difference in those powers of 10 that I'm showing you here, right? In this case, we need to calculate 2.5 million time steps in order to run the complete simulation. And this is why we really need to use GPUs for this process, because you can't parallelize CPU cores into the future. You can't create some workflow where some of the CPUs look at the time equals 0 events and some of the CPUs look at the time equals 1 events, because in order to understand what happens when time equals 1, you need to understand what happens when time equals 0. So the only way you can make these simulations faster is to use a more powerful processor, to use something that's just going to loop through that cycle really, really, really fast. And that's what GPUs are good for. And that's why we need to use GPUs to look at timescales on the order of 5 nanoseconds. So in terms of, just to give you a sense of, what these computational costs are like, I have a graph of the amount of nanoseconds for two different systems that I looked at on both carbonate as well as Big Red 200. And you can see that we're getting, somewhere in the realm of a half a nanosecond per hour to almost a full nanosecond per hour using our molecular dynamics engine of choice, which is OpenMM. So OpenMM, this is a Python package, completely installable with Anaconda, very easy to port over to Big Red 200. I will say that normally OpenMM runs a lot faster, but we're using a lot of custom code that slows us down by approximately a factor of 4. I'd also like to point out that the A100s, as you would hope, are faster than the V100s. We see a consistent trend of the A100s churning through the simulation at about an 18% faster rate. So how do you use these molecular dynamics to actually calculate that binding free energy, that physical quantity of how much, how thermodynamically favorable it is for a small molecule to bind to a protein? And you might think that you could just do that calculation directly. You might think you could say, hey, let's just look at the molecule in solution, look at the molecule in the protein, look at the difference in energy between the two and get some absolute binding free energy. In practice, that's really difficult to do because there are a lot of conformational changes that occur when a small molecule binds to a protein. So instead, the most common approach is what's called a relative binding free energy pathway. So now what we're doing is instead of looking at absolute, this small molecule going into the protein, we're taking an existing complex of a small molecule bound to a protein, and we're changing it into a different molecule. And if we do that both when it's bound to the protein and when it's not bound to the protein, when it's solvated, we're going to be able to calculate what's called a delta G of binding. And if you know what the free energy of binding is for one of these molecules, so in this case, if we know what the free energy of binding for L1 is, we can use this delta G to figure out what the free energy of binding is for the other molecule. We can use that to figure out L2. So this is much easier from a computational standpoint because there are less changes involved. And it also aligns really well with the goals of drug discovery because typically when you reach a stage in the process where you want to be using free energy calculation, you already have a molecule in hand that you're trying to sort of just make better. So you already have that L1 with a known binding affinity that you're working with, so you're already trying to improve an existing compound anyway, which is exactly what these calculations are doing. So in order to slowly change one molecule into another molecule, you actually have to run many different molecular dynamics simulations to do that. So you do what's called lambda scaling, where you have this value lambda that's going to detail sort of like the mix of how much of one ligand versus the other ligand you're looking at. And you're going to run a bunch of simulations where you slowly increase that value of lambda, where you're decreasing the input from one ligand and increasing the input from another. If we went back and looked at the equation for our force field again, really all we've done is just by numerical scaling. Since we already have a mathematical equation for how things interact with each other, if you scale some of those things to 0, that's like they don't exist. And if you scale, one to 30% and the other to 70%, you can get a lambda value of 0.3. So this is a very common way to do free energy calculations. It goes by a lot of names, and the names are mostly based off of the math that you do after the fact when you do the simulation, but all the computational cost is really on this side. It's really on running, in this case, 11 different simulations. So I'm going to call it thermodynamic integration. Right? So the problem with thermodynamic integration is that the number of simulations that you have to run, it starts to scale very quickly. Just to get a single binding free energy in this case, you need to run 11 simulations. If you want to run 5, now you need to run 55. So this is why we wanted to move away from carbonate to Big Red 200. Because carbonate has a user limit for the number of GPUs that you have. You can only use up to 4 nodes at a time, which means that your maximum GPU capability is 16 GPUs. And we were submitting jobs where we were asking to do like somewhere in the realm of 200 to even 400 simulations in sort of these burst workflows. We weren't necessarily running them constantly, but sometimes we would need a lot, and then we wouldn't need a lot for some time afterwards. And carbonate isn't really good for that because you have that user limit. Those workflows could take multiple weeks to actually finish. But in Big Red 200, because you have so many GPUs that are available, and because you don't have any user limit associated with your use of the cluster, you can submit jobs, have them all go, and then have them finish by the end of a weekend. Right? I've run workflows personally where for a short period of time, I was using somewhere in the realm of like 80% of the capacity of Big Red 200. And that really helps when you have these burst workflows that you need to do. So now I'm going to talk a little bit about the research of what the Vilsack group does. You might think, what are you using these actual GPUs for? What specific problem are you working with? And the truth is, we were using them for benchmarking for a new method of free energy calculation that we're developing. So in the Vilset group, we developed this new method called LaDyBUGS. LaDyBUGS is what's called a lambda dynamics method, where you look at multiple different small molecules simultaneously, constantly changing from one molecule to a different molecule, and that lets you get the information of many simulations, in this example, 55 simulations, in a single simulation. This can allow you to drastically reduce the computational costs associated with free energy calculations. So, the way this works is we set up this system of discrete lambda states. So I have this chart of, essentially dotted lines from each molecule to each other molecule. And what I want to point out is, along those lines, we're dealing with essentially the same lambda state that you're dealing with in a single TI instance. Granted, we're looking at a fully connected interface rather than what you might call a star map, where just one thing is connected to everything else. But that ends up being beneficial because that means that you can remove a lot of hysteresis effects. So just to give you guys a workflow for how this process works, we start by running a short molecular dynamics trajectory. We then use the updated positions of our simulation to recalculate the energies of all the different lambda states that we're looking at. We then use a bias to constantly push us from looking at one lambda state into a different lambda state. And we can update that to sort of discourage us from visiting states that have been previously visited. And then using that information, we do what's called Gibbs sampling and choose a new lambda state that we want to sample. So we're going to loop around this process a lot, but sometimes we take a break from that. We use the M-bar algorithm to actually calculate what the differences in free energies are between each of our lambda states, and use that information to update our biases some more. So like I said beforehand, we did benchmarking of both thermodynamic integration as well as LaDyBUGS. And we had 3 goals in doing this benchmarking. First, we wanted to show that LaDyBUGS could estimate the free energy of binding with an error of roughly 1 kcal per mole. 1 kcal per mole is what's considered the gold standard in computational chemistry for the level of precision that you can get in your analysis, because that's roughly the level of precision that you can get experimentally as well. Additionally, we wanted to show that our predictions were at least as good. They had very good agreement with what you could get with thermodynamic integration, because our goal wasn't necessarily to make a more precise method, but our goal was really to make a cheaper method. We wanted to show that our method was going to be substantially cheaper than thermodynamic integration. So these were the benchmark systems that we used. It was 3 different protein systems that used 24, I call them ligands here, but those are small molecules total. The LaDyBUG simulations we ran for 15 nanoseconds total. And for thermodynamic integration, we used 5 nanoseconds for each individual lambda window. And we ran all of these results in triplicate so that we could get some decent reproducibility with our data. So if we compare both of these methods to experiment, both LaDyBUGS as well as thermodynamic integration, I'm labeling that as TI/M-bar here, we get good agreement with experiment. We get root mean square errors that are in the realm of about 1 kcal per mole for both methods. But the result that we're really looking for, the exciting result for us, is that if we compare our method LaDyBUGS to thermodynamic integration, we get very strong agreement. The error here is now less than half a kcal per mole. But so these methods are giving you the same answer. The difference is LaDyBUGS is doing that with only 180 nanoseconds of total simulation time, but thermodynamic integration is doing that with over 3,000 nanoseconds of simulation time. So, if you crunch the number, that works out to be LaDyBUGS being, 19.2 times more efficient than thermodynamic integration, at least with the examples that I've shown here. So, just to summarize what I've talked about today, I talked about, how the drug discovery process works, and how, a key thing that we need to focus on is potency. And that really boils down to the physical quantity of that free energy of binding of that small molecule to the protein. I've demonstrated how GPUs provide an effective method to actually estimate that free energy of binding. I've shown you guys how Big Red 200 adds a lot of value to our workflow because it lets us churn through a lot more simulations than what we might do otherwise. And then finally, I've shown that, using the benchmarking that we've been capable of, or that we were able to do on Big Red 200, we could work on developing a new method that we've shown, is close to 20 times more efficient than the existing method of determining free energy calculations. So, in terms of acknowledgments, in academia, you tend to get a lot of pictures of like the group photo at the end, but the Vilset group is small enough that, that's just the 4 of us here. We did get a new postdoc that replaced me, but I don't have his picture there. So, just in terms of acknowledgements, I'd like to acknowledge both my advisors, Jonah Vilsack and Tim Richardson. Ryan Hayes and Xingchang Ding did a lot of help as well with the LaDyBUGS methods, but they're at different institutions. One's at MIT and one is now at UC Irvine. And finally, the Vilset group and the IU School of Medicine at large. So that's all that I have for you guys. Do you guys have any questions? Yeah. So the way we specify the number of GPUs is when we queue the job with Slurm. So we submit jobs in Slurm in an array, and we use that array value to sort of loop through different lists of variables. And then for each individual array job, we feed those variables to the code, and then that's going to look at the correct lambda state. So we're not using, so one of the advantages of thermodynamic integration is that you don't need to have those simulations talk to each other while they're running. So you don't need to do anything special with srun or anything like that. So we submit say 168 jobs to the queue, and they run whenever there's free space. But in order to get the results that we want, we take that data at the end and do a separate calculation. Yeah, did that answer your question? Okay. Yeah.
Hey, is it on? Okay. That was a great presentation. So
Thank you.
Can you back up to the summary slide for me? All right, so one of the main questions I have is your 19.2x efficiency.
Mm-hmm.
Is that on the same amount of compute power as your benchmark data that you ran on Big Red?
So we did this by nanoseconds of simulation. So, when I'm talking about efficiency, if you run stuff on Big Red, obviously it'll take less time to churn through a set number of nanoseconds than it would on, a V100 or a 2080 Ti. The LaDyBUG simulations were run on 2080 Ti's. We have a private cluster where we're doing that on. But the efficiency, that's all done by simulation time. So that's not looking at like necessarily, so this is not, so, that's not directly comparable to wall time because again, the platform you use, some can churn through simulation time a lot faster than others. And like I also said, because we're using custom code, that also slows us down. But these efficiencies are calculated by the simulation time. So, 19.2 is just 3,465 divided by 180.
Which is a great equation to have on any slide right there. So then with your OpenMM that you use for your benchmark to find AI, did you do any OpenMM with AI interface, or was it just a pure CUDA OpenMM?
This was pure CUDA OpenMM.
Okay. And is there any like next steps for LaDyBUGS in implementing like an AI predictor or even for the OpenMM, right? Because they're super speed up with
Yeah,
AI.
yeah, it is very sped up. So one of the things that's interesting about using a, sort of faster method of getting free energy calculations is that's just going to exhaust the ability of the human to come up with ideas very quickly. So, a methodology that's become very popular in the drug discovery community over the past couple of years has been to pair active learning processes with different methods of free energy calculation. So there are a couple of papers that have come out recently where you do active learning where you look at a set number of molecules with free energy calculations. You sort of learn from that experience and then use that information to predict what the next best batch of systems is, and then do that. And that lets you screen much larger sets of molecules than what you might be able to do otherwise. The challenge in implementing that with LaDyBUGS is that because we're looking at a lot of different small molecules simultaneously, there's some degree of choosing what small molecules to group together as well. So that adds an additional wrinkle to that existing workflow. But that's definitely something we're thinking about, because at the end of the day, if you can churn through these things very quickly, you want to start having a machine ideate new compounds rather than a person, because now you can just have that loop and have it just run.
Right. Okay, great. Thank you so much.
Yeah.
Folks, I'm going to pass the microphone around because we're recording. So if you speak in the room without using the microphone, the recording doesn't pick it up, or you can repeat the question.
Okay.
Okay. How far out of reach? You're doing one protein, many molecules.
Yes.
How far out of reach is one molecule, many proteins?
One molecule, many proteins would involve an absolute binding free energy. So that would be because these are relative binding free energy. So we're looking at changing a small molecule into a different molecule. So already that's two molecules, right? One molecule, many proteins. You can do absolute binding free energy calculations now. I would say they're maybe like 10 times more expensive than a relative binding free energy calculation. It's probably not doable today, but it's probably not as far off as you might think, especially with some of the advances I've seen in Hopper. Like the 4090 looks like you have like a 50% speed up in molecular dynamics over the 3090. So, that's a big advance on the hardware side. And even on the software side, people have gotten a lot better at doing absolute binding free energy calculations. And I think, sort of as I mentioned with Caleb beforehand, people have gotten pretty good at pairing these processes with active workflows. So you might imagine something similar with a large pool of proteins, and you use some active learning workflow to figure out, what molecule that protein is going to be better at binding to.
What was the number of GPUs in your project? It was limited or unlimited?
So we used one GPU per Lambda window in the thermodynamic integrations. So for this data set, we had 24 ligands. So that meant that you had 11 plus 5 plus 6. So we had 18 different TI calculations. We did it in the star map. So whatever 18 times 11 is for the TI side, that would be 198 GPUs. But then we did it in triplicate. So.
Okay, and you have the option to determine the number of GPUs in your project, or in general, it is unlimited?
Big Red 200 doesn't have a user limit for the number of GPUs you can use at any given moment.
Yeah.
There is a limit on the number of GPUs available, which I believe is 256.
Okay.
But that is, that's a pretty high bar, because these things will take, for 5 nanoseconds, that'll only take 10 hours per GPU. And, sort of as I explained earlier, the workflow, this is very easy to parallelize because you don't need the individual simulations to talk to each other. So you can run them, so you can run a batch of them, have them be on the queue, and then you lose priority, and then other people do their stuff, and then you regain priority, and then you can just keep going.
So the number of GPUs change from time to time. So the available number of GPUs.
The available number of GPUs on Big Red 200, yes, you would expect to change from time to time.
Okay. Okay. Thank you. Thank you.
Yeah.
Shall we thank Michael? Thank you.